Welcome folks. In this video we are going to focus on biology paper 2 for the year 2020, and we are dealing with a paper that was written by the internal candidates. Now let's first begin with questions from section A. So we have table 1 shows cells and tissues found in living things. Now let's look at the first row. So we have this row 1 here has to do with the cells. Then we can see that the cells that we have we have red blood cell, root hair cell, palisade cell, and sperm cell. Then the second row has the tissues, where we have xylem tissue, nerve tissue, epidermal tissue, and muscle tissue. Then not only should we know the type of tissue, then we also have to look at the function for each cell and each tissue. So if we were to summarize some functions of these cells and tissues, like for red blood cells, we have transportation of oxygen and carbon dioxide. Then if we look at the root hair cell, we have absorption of mineral salts and also water from the soil into the xylem. Then we have palisade cell. This is where the process of photosynthesis is high. Then the sperm cell has to do with reproduction and also carrying of genetic makeup from the parents to the offspring. Then if you look at xylem tissue has to do with the transportation of mineral salts and water from the soil to the leaves. Then for nerve tissue has to do with the nerves. If you look at the nerves, we are looking at structures where the nerve impulses are being transported. Then if you look at the epidermal tissue, it is coming from the word epidermis, that has to do with the protection of certain parts from mechanical damage and infection. Then muscle tissue has to do with movement. We know that if we are trying to move, bend, or maybe lift something, muscles are responsible for such actions. Now let's dive in for the first question. From table 1, identify a plant cell. So we can see that we have two plant cells here. The first one, so root hair cell can be the answer here, and palisade cell. Then for the second one, animal cell, so red blood cell, and the sperm cell. Then for Roman number 3, a tissue whose function is to cover and protect plants from mechanical damage and infection. So we know that that's epidermal tissue, because the function of the epidermis does the protection from damage and the infection. So it is epidermal tissue. Now let's look at the question for a tissue whose function is to transport water and dissolved mineral salts in plants. So it's a xylem tissue there. Then let's move on to question 5. A cell found in the reproductive system. So from our table, we know that we are dealing with a sperm cell. Then let's move on to question B. In which tissue are the following cells found? Now, so for us to answer the questions for B, we just have to look at the biology in general. So it's away from the table. So red blood cells are found in blood tissue. So remember, blood is a tissue because it consists of certain cells. Then palisade cell, the tissue in which palisade cells are found is palisade mesophyll tissue. Then we move on to question C. Name one organ found, the first one, in a plant. So there are so many organs. The first one, we know that roots are organs. Then stems also, they are organs. Even leaves are organs. Then in animals, we have like the kidney is an organ. The heart is an organ. Brain is an organ. Skin is also an organ. So there are so many organs, so of which you just have to choose the one that you are able to remember as soon as possible. So now let's look at the question 2. Question 2, we are also given a table. So table 2 shows the concentration of various substances in blood, plasma, glomerular filtrate, and urine. So we are dealing with the excretion, and whenever we look at excretion, we are looking at the process or processes that are involved for the body of a living organism to remove the waste products. So the waste products are things that are not supposed to be in a living organism, for they can interfere with the life processes. So the waste products, we have urea, we have uric acid, we have excess water, we have excess salt, then we have even ammonia. If you look at the skin, we have sweat. Then so there are so many, even carbon dioxide is also considered to be a waste product, so it has to be excreted. Now there are organs that you need to first have in mind that deals with the process of excretion. So the system, we know that the system is excretory system. Now under excretory system, we have excretory organs that are responsible. So if you look at the body, let's take it for instance, the body of a human being, you would find that each system has a role to play. Then if you look at the combination of the systems forms what we call an organism. So if you look at excretory system, it's a system that deals with the excretion, and the organs that are involved in excretion are known as the excretory organs. Then we have reproductive system that is responsible for reproduction. We have the nervous system that is responsible for coordination. We have endocrine system that is also responsible for production of hormones, which is also coordination. Then exocrine system that has to do with the formation of digestive liquids. Then we also have digestive system that is responsible for digestion. Then these are some of the systems that I've mentioned. Now the key point is, as you are studying biology, at last you need to come to understand that you should be able to connect and to see the connection, how these systems are truly connected. So these systems, they make sure that they work together so that life is possible. Now let's look at here the table. So the first column will have substance. The first one is urea, glucose, salt. Then we have protein. Then here we have blood plasma. So in blood plasma, urea, there is 0.04, 0.01 glucose, salt 0.46, proteins 8.0. Then in glomerular filtrate, so if you just look at the structure of a nephron, we know that the blood vessel that brings blood to the Bowman's capsule, this is what we call the afferent vessel, and this structure is what we call the glomerulus. Now here there is a cup-like shaped or a funnel-like shaped, and this is known as a Bowman's capsule. Now, under the Bowman's capsule, the function of the Bowman's capsule, it collects the glomerular filtrate. So the glomerular filtrates, these are substances that are forced out of the glomerulus into the Bowman's capsule. So whenever these substances, this may be urea, a small amount of glucose, salt, excess water. So whenever these substances are collected in the Bowman's capsule, they are now known as the glomerular filtrate. So this is the point here. At this point, it is not known as the urine, because we still have some processes that should undergo for us to have urine. Like we need to have selective reabsorption, where some substances that are still useful must be reabsorbed back into blood, and also we need to have osmoregulation. Now, after we have this, we can see that in terms of the protein in this glomerulus here filtrate, we don't have the protein. Even in urine, we don't have the what? The proteins. So we can conclude here that proteins cannot be filtered out. And the reason behind this, we know that in terms of the sizes, because there are only small pores here that can only allow small substances to pass through into the Bowman's capsule. Now let's look at the first question. Question A, Roman number 1. Name the process responsible for the formation of glomerular filtrate. So the process that I once looked at here. So what process is this one? So this is known as the pressure filtration. Sometimes you may find it as the ultrafiltration. It's the same. So why do we have pressure? So this means that for substances to move from the glomerulus into the Bowman's capsule, there should be that squeezing, there should be that pressure that should force the substances to fall into the Bowman's capsule. Then Roman number 2, which of the substances in table 2 above does not pass through the glomerulus during filtration? So here we have proteins. Then give a reason for your answer above. So the reason that we can give here is we know that because in glomerular filtrate and in urine there was we can't see the presence of proteins. So say proteins were not found, in glomerular filtrate and urine. And the reason behind that is because of the sizes of these proteins molecules. Now for question B, explain why the concentration of urea in glomerular filtrate is much less than the concentration of urea in urine. So let's look at in a glomerular we have about 0.04. Then in urine we have 2.0. So the reason behind this is because for urine to form, there are processes such as selective reabsorption must also be done. So by the time some of the useful substances have been reabsorbed back into blood, by then the only substance or substances that remains are the urea and just the waste product that are forming the urine. So that's the reason behind that. So say because all the useful substances have been reabsorbed into blood. So as a result, an increase in concentration, of urea in urine. Then for question number C, outline the internal structure of a kidney. So we want to look at the internal structure of the kidney. So how do we outline this? So the first one, we need to know that the kidney is surrounded by a fat layer. So that is the first point that we are going to look at. So the kidney is surrounded by a fat layer. Then the function of this fat layer is to protect the kidney from mechanical damage or any external damage. Then after this, we have what we call the tough fibrous membrane called renal capsule. So after this follows a tough fibrous membrane known as renal capsule. So we won't look at the function of this renal capsule. We are just looking at the internal structure of the kidney. Then below the renal capsule is the cortex. Which contains blood vessels. Then after the blood vessels, the cortex, we have the medulla, then we have also renal pyramids and renal pelvis. So you can add this. So we are saying after the renal capsule is the cortex, then inside the cortex we have the blood vessels that supply blood and also take away blood from the nephron. We know that the function of the nephron, that is now where the processes that are involved in urine formation occurs. So after this cortex, there is the medulla, then there is also renal pyramid and renal pelvis. Now let's move on to question 3. So question 3, figure 3 shows stages in the life cycle of a housefly. Then we have stage S, P, Q, R. So we can see that the first one, a housefly has 4 stages. Then the first stage, or maybe we can say we'll begin with the first one, adult. Then after this we may have eggs. Then after eggs we have larva. Now sometimes or in some textbooks you may find that larva may be divided into sub stages, where you may have the first larval stage, second larval stage, and the third larval stage. Then the last one, we can now look at the pupa stage. So the best way to remember the order of these stages, you just have to look at the alphabetical order. So you just have to alphabetize these letters. So you can see that the first one, according to the alphabetical order, is adult. Then we have E, L, and P. Now from this, we can be able to identify. Now P is simple. We can see that these are eggs. Then after eggs, we know that comes larva. Then after larva, we are going to have a pupa here. Then after pupa, there is this adult. Okay, then the other thing that you also have to know, this is what we call a complete metamorphosis. So this means that there are 4 stages that are involved. Then in complete metamorphosis, you may have 3 stages. So whenever you have a life cycle where you have less than 4, that is incomplete metamorphosis. So the type of metamorphosis that in which a housefly and mosquito undergoes is complete metamorphosis. Now let's have the first question. The first question is identify stages P and R. So P, we can see we have eggs. Then R is pupa. Then let's move on to B, Roman number 1. Which stage is useful in the decomposition of matter? So decomposition of matter, we are looking at like decaying or to break an organic matter into simpler. Now, let's have one example of maybe a corpse of an animal or maybe a human being. We know that if someone dies, you may find a lot of houseflies on the body of that particular individual. And after that, you may find that the person will start to decay. So the stage that is involved in decomposition is just an adult. So that is, according to the given, we can see that that is S. So adult is involved in decomposition. So S, which is adult stage. The best example I've explained, if you look at the corpse. So it is the adult houseflies that you may find there. Then Roman number 2, which stage is influential in the spread of diseases, is still S. Because whenever we have an adult, that adult is now be able to move from one position to another. So as it is moving from one position to another, it is carrying some of the pathogens that causes depends on the type of disease that you are looking at. Remember, a housefly is a vector. This means that it transports certain pathogens that causes some examples of diseases, like if you look at cholera and also dysentery, even typhoid. So a housefly is a vector for such kind of diseases. Then state 2 diseases of which a housefly is a vector. So I've mentioned we have cholera, then also we have dysentery, even typhoid is an example. Then we move on to question D. Explain how a mosquito is similar to a housefly in terms of the first one, life cycles. So the first one, remember I mentioned earlier that the mosquito and the housefly, these two undergoes the complete metamorphosis. So they undergo complete metamorphosis, which is 4 stages. Then Roman number 2, spreading of diseases. So we say a mosquito is also a vector, meaning it has the ability to transport or transmit some pathogen. Then let's go to E, where we have explained one way by which a housefly can lead to the spread of cholera. So now we are looking at let's say, for example, there is a dump site, and also maybe you have food that is uncovered. So what happens is, if a housefly goes onto that substance, that on a decaying substance or waste substances, so if it sits on that, when moving from that into the food that is supposed to be eaten by an individual, it carries its pathogen on their legs and also on the small hairs that covers their bodies. So we'll say this, we'll explain this. So we'll say houseflies carries pathogens on their legs and on the small hairs, that cover, so say that cover their bodies. So this means that the pathogen, like the Vibrio cholerae, which is the pathogen for cholera, will stick to the legs of a housefly and also to the hairs that covers the body. Then as it moves from a substance that is decaying or a substance that is rotten to maybe a plate of food that is uncovered, then the pathogens will be transmitted in such a manner. Then also the key point is a mature housefly will use the saliva to liquefy solid food before feeding on it. So as it is using its tongue to liquefy the solid before eating, then upon doing that, when moving from that food onto some food that is uncovered, then the pathogens will be transported in such a manner. So that is, so the whole thing, maybe to summarize or to make it simple, we are saying if we have something that is rotten or something that is considered to be a waste product here, and we have a plate maybe of food that is not covered. So if a housefly moves from here up to, if it sits here, when moving it will carry some pathogens. We are saying some pathogens on the legs and also on the small hairs. Then if it sits here, it is going to drop some of these. As a result, when a person eats this food, then that person will now develop the cholera disease. Then let's move on to question 4. Figure 4 shows structures of two neurons. We know that two neurons, we are looking at cells that were created for transportations of nerve impulses. Then if you look at nerve impulses, definitely we are looking at the information that is responsible for response of a human being to any external change. Now let's look at neuron 1. So neuron 1, we can see the direction. So if you look at this, as long as if you have studied biology carefully on the nervous system, you know that these may be the muscles or sometimes maybe the glands. Then muscles are examples of what? The effector. Effector is an organ that is responsible for an organism such as an animal to respond. Then we have these, which we are referring to as dendrites. Then we have our Q. Our Q, we call this one as the myelin sheath. Then we have the line, the passage, which we call the axon. Then we also have the space in between these myelin sheath, and this is known as the nodes of Lanvia. So we have nodes of Lanvia. Then the myelin sheath are made from the cells that are known as the Schwann cells. Now, so whenever we have the arrangement of this, so whenever you have muscles or whenever you have an effector and the cell body, this is where the cell body is. And you just have to look at the location of the cell body. You'll be able to see the difference between this neuron 1 and the other sensory neuron, because this neuron is a motor neuron. So if you look at the sensory neuron, the cell body is located like this. This is how you find the cell body. Then you have these cells like this. So I'm sure you'll be able to see the difference between these two structures. So we know that neuron 1 will be motor neuron. Then for neuron 2, this is a relay. Relay has so many names. You may find it as multipolar, connector, intermediate, or these are the names of this. So for the first one, A1, identify two neurons shown in figure 4. So the first one, we know this is a motor neuron. Then the second one, we have a relay neuron. Then to state the functions of parts P and Q in neuron 1. So let's go back to neuron 1. So P we know it's dendrites. So the dendrites, or this is the dendrites, is a part in between the junction between two neurons. And the dendrites for a motor neuron, it's a part that it uses to receive electrical impulses from the neighboring cell. So we'll say these are parts that are used to receive electrical impulses from the neighboring cell. So from the neighboring cells. Then let's look at Q. What is Q? Q, we said myelin sheath. So myelin sheath, it is made up of a fat layer also. Then the function, we can say it acts as an insulator. So the simple language that we can use here is, we know that if we have two neurons like this, maybe the first one is carrying the impulses in this direction, then the other one is carrying the impulses in an opposite direction. So we don't want if this is our neuron 1 and this is our neuron 2, so we don't want the impulses from crossing. We don't want impulses to cross from neuron 1 into neuron 2. So that is the function of this myelin sheath. So say it prevents electrical impulses from jumping, so from jumping into another or into a neighboring cell. So that is the functioning of this structure. Then if we go back here, we'll look at the function of marine sheath, which is acting as an insulator. Then nodes of Ranvier, the function of these parts, it is just there to accelerate or to increase the speed of electrical impulses. Then we also have the axon, which is the passage for electrical impulses. Now let's go to the other part. So the other part will have B1. So under B1 we have B1. What do you understand by the term reflex action? So the simplest definition that we can give for a reflex action, this is just an automatic action, or we can say it is just an action in which we do not participate in terms of thinking. It occurs without our consent, and most of these actions are there to protect us from danger, because it would have been taking long for an impulse to move from, let's say, from the receptor then into the sensory up to the brain. So if the interpretation were to occur for every action, it would have been taking so long. As a result, then we may encounter some mechanical problems. So to prevent that, we need a fast response, a response that may or that should protect us from any danger. So that's why we have the reflex action. So we'll say this is an involuntary immediate action to a stimulus. So a stimulus is anything that may cause an organism to respond. For example, when it's cold, we have to make sure that we put on something that is hot. So the temperature, the reduction in temperature is a stimulus, because that has caused us to respond to change in temperature. Then we can move on to Roman number 2. So Roman number 2, we want to give one example of a reflex action. Actions. What are the examples of actions that you think they occur automatically? They are automatic in terms of occurrence. So the first one, we have blinking of an eye. So if, for example, an object is approaching an eyeball, so you need to blink to prevent that from entering or to cause damage to the eyeball. Also for lubrication in terms of tears. Then the other one, we have the knee jerk is an example. Then also we'll say withdrawing. So withdrawing a hand from a hot object. Swallowing is also a reflex action. Then coughing is also a reflex action. Then for Roman number 3, describe the path taken by an impulse from the receptors to the effectors. So maybe let me see if I can give you the picture of this. So the first thing that you need to know about the reflex acts easy. The first one, we need to have a stimulus. So let's say you accidentally touch something that is hot. So say a hot object. So this hot object will be considered as a stimulus because it is going to cause you to respond. Then after the stimulus, we need to have the receptor organs. Then the receptor organs, these are the organs that we consider to be as 5 senses. So if you accidentally touch something that is hot, the receptor will be the skin because it is going to be this is the organ that will be in contact with a hot object. Then the other receptor organs that we can look at apart from skin, we have eye. Then the stimulus for an eye is light because we know that we respond by light. Then after eye, we have the E, where we have the sound. So this is the stimulus for ear. Then after that, we have the tongue, which is also a receptor organ, which is stimulated by taste. Then we also have the nose, which can be stimulated by the smell. So these are receptor organs. So after the stimulus, we need to have a receptor organ. So a receptor organ, the word receptor is coming from the word to receive. So these are organs that are receiving the changes from the stimulus. Then after that, we are going to have sensory neuron. So we know that these neurons now will transport the electrical impulses from the receptor to the central nervous system. Now the central nervous system, we know that these are known as the coordinators. Then under the coordinators of the central nervous system, we have the brain. We know that some interpretations occurs in the brain. Then we also have the spinal cord. So the spinal cord and the brain are collectively known as the coordinators, which is the central nervous system. So in the central nervous system, this is where the relay neurons are found. Then from there, we are going to have the motor neuron. So the motor now will transport or will carry the information that is being transported. Then from the motor, you are going to have the effector. So the effector, we can look at like muscles. So for you to move the hand from that object, the muscles must contract and relax. So there should be that antagonistic kind of action. Then if you withdraw the hand away from that hot object, this is what we call the response. So from here all the way up to the response, this is what we call the path taken by an impulse from the receptors. Now if you look at a relay, so you need to view a relay like some of the sports that you've once undergoes. There is that running race where you have maybe a baton. So one is carrying a baton, then if reaches another line where there is maybe a group of people belonging to that group, then that person will get that one who keeps on running. So this is what happens. So it's a relay. This means that it takes the information from the sensory, then it gives it to the motor neuron. So that is the key picture that we can share for this one. Now we move on to question 5A1. State the number of sex chromosomes in the nucleus of a somatic cell of the human body. So the total number of chromosomes for a human being under someone who is normal is 46. Now, the way this 46 is you have to remember that the 23 chromosomes came from the male parent, because we know that here we are going to have sexual reproduction, and whenever we have sexual reproduction, we should have two parents. There should be a male and the female. Then the other 23, these are coming from the female parent. So we can see that now the total number of chromosomes, 46, is just the sum of the half from the female and the other one from the male. Now, if you look at this, so let's consider this one as an ovum. So an ovum is just the gamete or gametes that are produced by the female. So in this 23, there should be 22 that we call the autosomes. Then the other one must be the sex chromosomes. So let's say this is the X. So in this 23, we are going to have the same. There should be 22 autosome plus let's say if this is X. So I want you to see something here. So the sperm cell may carry an X, or sometimes it may carry an Y. So that's a pattern. But for a female, we know that every ovum, according to science, carries XX. So the determination of sex is all about which ones of these two X chromosomes are going to meet. If, for example, we have 22 plus X, so let's say the sperm cell that is fusing with an ovum is carrying X. And if you draw a genetic diagram, we can see that this X will mate with this X, will give us XX. And we know that whenever we have XX, this is giving us that this is a girl child. Then let's say the sperm cell that fuses with the ovum is carrying the Y. Remember, the ovum will always carry an X. So in this particular point, we are going to have XY. So the determination of the sex of the child here can be simplified as it depends on the chromosome that is being carried by a sperm cell, because the ovum, according to science, will always carry an X chromosome. So now the point here is you can see that if this sperm cell fuses with this ovum, this 22 plus this 22 will give us 44. So there are 44 autosomes chromosomes. Then this, let's say X plus this X, we are going to have XX if this is a female. So you can see that in a somatic cell, there are two sexy chromosomes. So here the answer is two sexy chromosomes. Now remember, if we are looking at a lady, the two sex chromosomes for a lady will be XX. Then if it's a man, we are going to have XY. But the key point is we just have to have two sex chromosomes under normal circumstance. Then we have Roman number 2. So Roman number 2, we are taught to define or maybe explain what hemophilia is. So hemophilia, we are looking at a blood disorder, and this blood disorder, it is just a disorder that affects blood. How? It's a disorder in which the blood cannot clot normally. So that's a point. And the reason why this may be there is because of there may be insufficient or there may be low clotting factors. And some of the symptoms, that is or that are associated with someone who is hemophilic is that the bleeding takes time to stop, or sometimes impossible. As a result, they lose excess blood. So here, the point for hemophilia will say this is, remember, this can be defined in so many ways. So this is a blood disorder. Blood disorder in which blood does not clot properly or normally. So say due to low clotting factors. So here you just have to look at maybe some of the factors that helps blood to clot. So you just have to, if you want to look at that, you can go and look at the mechanism during the clotting of blood. Now here we have question B. Using a genetic diagram, explain how normal parents for hemophilia can have a son who is hemophiliac. Use symbols XH and X small letter h to represent the dominant and the recessive alleles respectively. Now maybe before we dive into this genetic diagram, there are some stuff that I want you to know. So let's first begin with a man. So a man is either that man is okay or sick. There is nothing like a carrier for a man. So now if the man is okay, you only put a capital letter on the X chromosomes. Remember, do not put anything on the Y chromosome. Why? Because the Y chromosome does not carry these genes that carries the disorders. So this one indicates that the man is 100% normal. Then for a man who is hemophiliac or a son who is hemophiliac, you are just going to put a small letter h here. So this one is just a patient. Then let's look at for a woman. So for the woman, if the woman is 100% okay, we know that we are going to have XX. So if the woman is 100% okay, we are going to have two letters X, Y that are capital letters. So we need to have a uppercase. So this is 100% normal. Then the second one, we are going to have a capital letter H, and the other one, we are going to have a small letter h. Now, this woman will be considered to be a normal, but remember, this person is a carrier. So for a person like this one, the mother may not be a victim to this, but the problem is this disease may affect her children, especially a son. Now for a woman who is 100% sick, or we'll say a patient, for a patient the genotype will just be the small letters here. So this is what you need to have, and this information that I've shared with you guys can be used to explain or draw a genetic diagram for any blood disorder. Now let's go back to our question. So the question is, a normal parents. So there is something that I want you to commit to memory if possible here. Whenever you hear that a normal parent had a son who is hemophiliac. So if you look at normal parents having a son who is hemophiliac, just know that the mother is the carrier. The husband will be okay, because we don't have carrier if you look at the male part. So let's show what we are saying here. So whenever you hear a question like a normal couple, just know that the woman is the carrier. And you know that the carrier, we are going to have the capital letter and the small letter. Then since this is normal, just know that the man will be 100% okay, since a man cannot be a carrier. So we refer these as genotypes of F1 generation. So let's have random fertilization. We separate these. Remember, there are so many ways in which you write genetics, genetic diagrams. So please stick what you think is okay for you. The concept, as long as you keep the concept. So let's look at this one here. So this is showing us that this couple, so these two parents have a chance of having a daughter who is 100%, okay? Remember, these are just chances. Then they also have a chance of having a daughter who is a carrier. So the other point that I would like you to jot down is, if a man is okay and a woman is a carrier, they cannot have a daughter who is sick. They can only have a daughter who is a carrier. You can see by this genetic diagram. Now let's look at the other one. Here we are going to have, they have a chance of having a son who is 100% okay. Then they also have a chance of having a son who is hemophilic. So you can see where this problem is coming from. That's why if a man is okay and a woman is a carrier, there is the chance of having a child who is sick is just for a son. A woman, they cannot have a daughter who is hemophilic. According to this, that is impossible. So say genotype of F1. So this is what I can share with you for this question. Now let's move on to question 6 from section B. Now here we'll do the same. I just want to we'll just have a discussion over some of these questions. So the first one, question 6A, explain the stages involved in a testing piece of onion for reducing sugar. So what are we supposed to do? What are the steps that must be there if we are testing for reducing sugar? So the steps, the first stage. So the first stage that must be there is you need to, we will not focus on the materials that are needed for now. We just focus on the method. So the first one, you have to make sure that you heat water in a glass beaker on a tripod until the temperature is at 40 degrees Celsius. Then after this, it depends with the way an experiment is done. But if you want, you can label the test tube according to the amount of the food substance that you are trying to test. So let's say you label the test tube. You say test tube, you can just label it maybe A. So label the test tube A with the content that you are trying to test. Then number 2, what do? You add about let's just say 2 mils water and half a spatula of what substance are you trying to test. So we know that we have, this should be a grinded onion or a liquid onion. So you can just maybe add this and half a spatula of pounded onion. Then after that, what are we supposed to do? You need to now add at least 2 drops of Benedict's solution. Now remember, this solution is blue in color. So after you do this, you have to make sure that you swirl the test tube so that the mixture mixes. Then after that, place the test tubes in the water bath. Then from there, you just have to observe and record any color change. Now, let's say we have some color changes. So the first one, we know that we are beginning from blue, the color of the appendix. So if this changes from blue maybe up to green, sometimes it may just give you up to green. What is the the conclusion? So whenever it gives you a green color, just know that reducing sugar is present, but in a low concentration. It's there, but in a low concentration. But if it changes from blue to green, then after that it may change to orange. If it reaches orange, know that reducing sugar is present in a high concentration. Sometimes it may reach up to brick red. So whenever you have brick red, then just say reducing sugar is now present in a high concentration. So if the color changes only up to green, you have a low concentration. Then if it reaches orange, high concentration, brick red, high concentration. So that is it. These are the points that you have to put whenever you are asking a person to look at the testing piece of a substance for reducing sugar. Now let's look at B. Explain the following nutritional disorders. So if you look at a disorder is just a condition in which a certain part or organ of a living organism is not functioning properly. Now let's look at goiter here. So goiter can just be defined as the swelling of this can be thyroid gland. So if you look at the thyroid gland and the hormone production, we know that this is endocrine system. The function of thyroid gland is to secrete the hormone known as thyroxine, which is responsible for basal metabolic rate. So if for this gland to swell, it is due to the low, or sometimes we can say absence of an element known as iodine. So the point is, if the question comes, which substance must be absent for a goiter to develop? So it has to be the iodine. Then if you look at kwashiorkor has to do with where the growth of a child is retarded. Retarded here, we are looking at where the processes that are responsible for growth are very slow. You may find out that someone who has this kwashiorkor, may the body may not be correlating with the age. You may find that someone who is maybe 10 years of age is appearing as if that person is maybe 5 years of age. Now, in terms of what substances must be lacking for this person to develop, mostly it is all about the proteins. So if you look at the absence of proteins in a diet, especially in children, may result in a condition known as kwashiorkor. Then if you look at the leaf chlorosis, this has to do with the plants, and it's a condition where the leaves turns yellow. So we are saying this is a condition also where we can see that there is insufficient of chlorophyll, so there is no that green pigment. Now, what can cause this? So this is caused by the absence of magnesium. So the function of magnesium, one of the function of magnesium in plants is there to make sure that the chlorophyll is produced. Now let's move on to question 7. Under question 7, we are looking at describe the process of reproduction in a frog. So there are some stages that you need to know about reproduction in a frog. So the first one, we know that if you look at frogs, the first step that you need to look at here, know that frogs reproduce in water. So the whole reproduction process occurs in water. Then the first thing that happens is the male frogs will call to attract the female, especially sometimes it depends on the area where you live. I'm sure during at night you may hear those sound of these frogs. So that sound is coming from a male frog, and that is the way of attracting the female. So once the male has attracted the female, then the male jumps on the female's back and holds her tight. So that's what happens. Then the presence of the male on the back of the female will induce now the female to lay eggs. Now after the eggs are laid, then the male frog then releases his sperm over the eggs. Then we'll see that here we are going to have what we call external fertilization. External fertilization, it is just the type of fertilization that takes place outside the body of the female. So unlike human beings, we know that with human beings we have the internal fertilization. The sperm cell will fuse with the female sex cell with female sex cell inside the body. So that is the difference that we'll look at, and we'll look at the difference for that. But the type of reproduction that occurs in frogs, we are looking at the external. So if you are taught to maybe explain or define external fertilization, we are just saying it is the type of fertilization that takes place outside the body of the female. Usually it occurs in water. So we are saying this is external fertilization, and it takes place in the water outside the female's body. Then the importance of water will prevent the eggs from drying out. Then the goal of a frog is to fertilize the maximum number of eggs in order to produce the maximum number of offspring. Now, most frogs are what we call oviparous. So if you look at oviparous, this means that the form of sexual reproduction in which the female lay eggs that hatch, so the hatching occurs outside the body. So remember, the type of fertilization is external. Also, the hatching is also external. So oviparous has to do with the hatching that is taking place outside. External fertilization is dealing with the type of fertilization that is also taking place outside. So these are points that I can share with you on frog. Then there is another term that I remember. There is what we call fecundity. So fecundity here, it has to do with we can see that reproduction output is high. So the number of eggs that are laid is high, but few fertilized eggs developed into adult frog. So this is what we call fecundity. Now we can look at B, explain how reproduction in frogs differs from that in human beings. So we just say in frogs, we have what we call external fertilization. So external fertilization is a type of fertilization in which the sperm or sperm cells fuses with eggs on the outside of the body. So that is what we have here. Whereas if we look at human being, we are looking at the first one. We are looking at internal fertilization because it occurs inside the body. So we have internal fertilization. Then here, no eggs will be laid under internal fertilization, but the other one. Then let's look at question 8. Describe the components of blood and their functions. So what are the components of blood? So the first component of blood, we can look at plasma. Now plasma can and is defined as it's just the liquid part of blood. That's liquid. Then in some textbooks, you may find it that this is a yellowish liquid, but don't worry too much on the color. Then the function, we know that this is where the cells will be suspended. So the red blood cells, white blood cells, even in platelets, these cells are found or suspended in plasma. Then the other function is for transportation. So plasma is responsible for transportation of the first one, blood cells. Then we have food values, we have waste products, then we have hormones. So to mention a few, these are some of the functions of plasma. Then the second component of blood is red blood cell. Now red blood cell or red blood cells, these are the cells that have no nucleus. And I'm sure you remember the reason why there are no nucleus. So to create a large space for hemoglobin. Then in terms of the shape, we say that they are biconcave in terms of the shape. Then the function is to transport oxygen and carbon dioxide. So to transport oxygen and carbon dioxide. Then the third one that we can look at is white blood cell. Under white blood cell, we have the cells that have lobed nucleus. Then there are so many examples of white blood cells. We have phagocytes, lymphocytes, neutrophil, so all these. Then the function of white blood cells are there to defend the body against infections. Then also sometimes you may also encounter like the name, the biological name for white blood cells. These are known as the leukocytes. Then for red blood cells, we have erythrocytes. Then the fourth component that we can look at here is we have platelets. Now platelets are considered to be just the fragments of cells. Then the main function is for blood clotting. So if you are able to expand these points that I've shared with you guys, at least you'll be able to get, at least if you are getting 10, 11 marks, that's okay. That's okay. Then we move on to question 9a. Explain the following types of growth with specific reference to plants. So growth is defined as the permanent increase in mass and in size. Now, what is what is primary growth, and where do you think this growth occurs? So primary growth, it is just the type of growth that involves the plant to grow in length, in height. So here we are not looking at the size or the diameter of the stem, we are looking at the height. So this one is responsible for the height or length. Then where does this process occurs? So this process occurs in what we call apical meristem. So under apical meristem, we have structures like the shoot system. So this is where it occurs, and the root system. So this is where primary is. Then we know that if you look at the seed we have structures like the plumule and radicle. So plumule changes into a shoot system, radicle changes into a root system. Now the process in terms of the process that is responsible for the primary growth to be there is where we have now cell division. Now cell division here, that is mitosis. We know that mitosis has to do with the multiplication of cells. Then after this cell division, we may have cell elongation, where the cells grow bigger in size. Then after cell elongation, we may have cell differentiation, in which now the cell becomes specified for specific actions. So remember, we are saying primary growth is the growth responsible in length, then it occurs in apical meristems. Here we have the shoot system and the root system. Then the process that is responsible is the cell division, which is mitosis, and the increase in terms of number of cells. Then we move on to secondary growth. Here we are looking at the increase in diameter. So diameter, it means if this is the size of the stem, the increase here, like if the plant is increasing in size in girth, so diameter, or we can say in girth, this is the secondary growth. And here we are going to have lateral meristems. That's where this occurs. And an example of a part that is involved in secondary growth, we have structure like cambium. Then we go to question B. Explain the differences between tropism and taxic responses in living organisms. So if you look at tropism, the first one, tropism is found in plants. Then this is the kind of response where a plant responds by bending. It can be bending away or toward a stimulus. So away, we know that away is negative for a certain party, then toward is positive. Whereas a taxic response, this one has to do with the animals. And here you just have to know that the entire body moves. So for example, if you as an individual, if you move from one position to another position, if you move from your home, let's say going to school, that is a taxic response. Now that is just movement, but let's say maybe you are sitting where there is too much sun and you feel like it's very hot, then you want to move into where we have the shade. So that movement away from the light into an area where you feel will be of much good temperature according to your need of your body. So that response where the entire body moves away from that is ataxic. And for example, the other best example, if you look at the behavior of cockroaches, whenever you switch on the light, you see the cockroaches, they move from the presence of light to a darker region. So that is another toxic because so under toxic, we are saying it is found in animals, then it involves the entire body to move. Then for tropisms, it is found in plants, and this involves only a certain party will respond. Then question 10a, explain the following terms. The first one, abiotics factors in an ecosystem. So if you look at ecology, ecology is just the branch of biology that tells us, or that helps us to understand the relationship that is between living organisms and non-living organisms. We living organisms depends on non-living organisms to survive. For example, we need air to survive, we need water to survive, we need the soil to survive. So that interaction in between these two organisms is what the ecology is. Now, if you look at the abiotics factor, abiotics factors, according to the biology, these are just non-living organisms. So organisms in which there is no life. So non-living organisms, these can be like the stone or stones, soil, water, oxygen. Then if you look at Nietzsche, Nietzsche is considered as a role played by an organism in its community. So the point here is every organism has a role to play in the community. That's why if you get rid of a certain species, then the ecosystem becomes disturbed. So there will be some organisms that will suffer because a certain species is removed. So the key point is, even as human beings, there are roles that we are supposed to play in our community, in places where we live. So the question that I'm going to leave you is, what is your role? What do you do in your community? What are you going to be remembered for when you die? What will people say? We miss this person. So that's the question that I will leave you guys to look at. So this is what I can share with you for this paper. Thanks for watching.